Homeostasis derived from the Greek word homeos or similar, and stasis or standing still, refers to the body's ability to maintain a stable internal environment despite of external changes. This involves a complex interplay of psychological mechanisms that constantly monitor and adjust various parameters such as temperature, blood pressure, pH, and also the blood glucose levels to keep them within a narrow optimal range. Maintaining homeostasis is crucial for survival, as even small deviation from these optimal ranges can have significant, very negative feedback loops, which counteract changes and restore equilibrium. Blood pressure is the pressure with which blood flows in the blood vessels. When the heart pumps blood into the systematic aorta or the pulmonary trunk, blood flows with pressure into the blood vessels. Arteries receive blood from the heart. Blood pressure is higher in the arteries. Blood pressure gradually decreases as blood flows into capillaries. When blood is collected into veins, blood pressure is greatly reduced because blood pressure is less in the veins when compared to the arteries. Blood pressure is measured using a sphygmomanometer. Blood pressure levels are written in the form of 120/80. This is normal blood pressure. The numerator is 120 represents systolic pressure, and the denominator 80 represents diastolic pressure. During systole, when the ventricles contract, blood is pumped into the arteries, increasing blood pressure. Immediately after systole, the systolic pressure rises, and the blood pressure in the arteries is called systolic pressure. When the diastolic occurs, blood vessels return to their normal diameter, and the maintain blood pressure at a lower level.
Insulin and glucagon, a balancing act. The human bodies maintain delicate balance of blood sugar levels. We have two K of hormones, the insulin and glucagon, play crucial roles in this regulation, working in opposition to each other to keep blood glucose within a healthy range. Insulin, the lowering hormones. When we eat, our bodies break down carbohydrates into glucose, which enters to the bloodstream. Raising blood glucose levels trigger the pancreas to release insulin. Insulins act like a K, allowing glucose to enter the body cells, where it's used for energy or storage, as a glycogen, in the liver and muscle or fats. This process reduces blood glucose levels, preventing them from becoming dangerously high. If the body doesn't produce enough insulin, as in a type A diabetes, or the cells don't respond properly to the insulin, as in a type 2 diabetes, blood glucose levels can remain elevated, leading to various health problems. Glycogen, the raising hormone. When a blood glucose levels drop too low, for example, meals or during exercise, the pancreas releases glycagon. Glycagon is a primary function is to signal the liver to break down stored glycogen back into glucose and its release into bloodstream. This raises blood glucose levels, preventing hypoglycemia, low blood sugar. Insulin and glycagon work together in a feedback loop. When blood glucose is high, insulin is released to the lower it. When blood glucose is lower, glycagon is released to raise it. This dynamic interplay maintains blood glucose within a narrow, healthy range. This image, the process of how increased blood glucose levels lead to a decrease in the blood glucose levels through the action of insulin. So first, increase blood glucose. The process begins within an increase in the blood glucose levels. This typically occur after consuming carbohydrates. Pancreas responds. The elevated glucose levels are detected by the pancreas, a vital organ in the endurance system. Insulin release. It responds to the high glucose levels. The pancreas releases insulin into bloodstream. Insulin, it is a peptide hormone that plays a crucial role in regulating blood sugar. cellular receptor. Insulin then binds the specific receptor located on the surface of liver cells, muscle cells, and other cells throughout the body. These receptors are essential for the actions of insulin. Glucose uptake. This bend of insulin to its receptor triggers increased glucose uptake from the blood into the cell. This means glucose moves from the bloodstream into the cells. Glycogen productions. Once inside the cell, the glucose is used for energy or stored as a glycogen in the liver and muscle or as the fat. Disposal. Glycogen is a storage form of glucose. Blood glucose decrease. As a result of glucose being taken up by the cells and stored as a glycogen or fat, the blood glucose concentrations decrease. This negative feedback mechanism help maintain blood glucose homeostasis. This image illustrates a negative feedback loop where high blood glucose stimulates insulin release, leading to glucose uptake by the cells and subsequent decrease in the blood glucose levels. This crucial process for maintaining healthy blood sugar levels and preventing hyperglycemia.
This diagram illustrates the process by which the body responds to low blood glucose levels, primarily through the action of glucagon. Here's a step-by-step explanation for you. The first one is decreased blood glucose. The process starts with a decrease in blood glucose levels. This can happen due to prolonged fasting, strenuous exercise, or other factors that deplete glucose stores. For the second one is pancreas and glucagon release. The pancreas detects this low blood glucose. In response, it releases glucagon, another crucial hormone in blood sugar regulation. For the third one is liver's rule. Glucagon's primary target is the liver. For the fourth one is glycogenolysis. Glucagon signals the liver to break down stored glycogen, or also known glycogenolysis. Glycogen is the storage form of glucose in the liver and muscles. For the fifth one is the gluconeogenesis. If glycogen stores are insufficient, glucagon also stimulates gluconeogenesis is a metabolic pathway where the liver converts non-carbohydrate sources such as amino acids and other molecules into glucose. For the sixth one, glucose release. The glucose produced through glycogenolysis and gluconeogenesis is then released into the bloodstream. For the seventh or for the last one, blood glucose increase. This release of glucose into the bloodstream increase the blood glucose concentration, counteracting the initial drop and restoring blood glucose levels to a normal range. In short, this diagram shows a homeostatic mechanism where low blood glucose triggers the release of glucagon, leading to glucose production and release by the liver, ultimately raising blood glucose levels. This is vital for preventing hypoglycemia or dangerously low blood sugar.
Regarding blood glucose levels, heat balance, and the thermoneutral zone. Let's begin with blood glucose levels. Blood glucose levels vary depending on timing and health conditions. For a person without diabetes, normal levels before breakfast are 70 to 99 mg per deciliter or 3.9 to 5.5 millimole per liter. Millimole means 1,000 of a mule. 2 hours after a meal, they are below 140 mg per deciliter or 7.8 millimole per liter. For a person with diabetes, levels before breakfast are 80 to 130 mg per deciliter or 4.4 to 7.2 millimole per liter. 2 hours after a meal, they should be below 180 mg per deciliter or 10.0 millimole per liter. At bedtime, normal blood glucose level should range from 120 mg per deciliter or 6.7 millimole per liter for people without diabetes. For those with diabetes, levels should stay under 150 mg per deciliter or 8.3 millimole per liter. Blood glucose levels are usually lowest in the morning before the first meal. They rise after meals by a few millimoles. Maintaining normal ranges is essential, as high levels can lead to hyperglycemia or also called high blood sugar. and low levels can cause hypoglycemia or low blood sugar. In temperature homeostasis, the average temperature inside a human body is 35.5 degrees Celsius to 37.7 degrees Celsius. Temperature homeostasis within the body is the balance between heat input and heat output. If you might not know what these two are, heat input basically means heat gained from the environment, for example, like a warm surroundings or a sunlight. While heat output is the heat lost to the environment through processes such as radiation, conduction, convection, and evaporation. Moving on, let's talk about heat input and output. Heat input can come from two sources. Internal sources, which include heat produced inside the body through metabolic process. External sources, which includes heat produced outside the body, such as the environment or artificial heating. On the other hand, heat output or loss occurs through several processes. First one is conduction. Heat transfer through direct contact with surfaces. The second one is convection. Heat loss through air or liquid movement. The third one is radiation. Heat transfer the form of infrared waves. The last one is evaporation. Heat loss through sweat, which cools the body as it evaporates. Neutral zone. The term neutral zone is the range of environmental temperature where the body can maintain its core temperature without expending extra energy. For humans, this range is around 55.5 degrees Celsius to 57 degrees Celsius. In this zone, when body temperature drops, blood vessels constrict to conserve heat. The body might also shiver, which generates heat through muscle activity. When body temperature rises, blood vessels dilate to release heat. Sweat glands secretes fluid, and as sweat evaporates, it cools the body. Maintaining a balance in body temperature is crucial for optimal physiological function. The body constantly adapts to preserve this balance.
The text explains thermoregulation and negative feedback mechanisms in the body. Thermoregulation, controlled by the hypothalamus in the brain, maintains a stable internal temperature. Negative feedback is the process that reverses deviations from a set point, keeping body parameters within their normal range. The example given is the glucose level control. Cells need a precise glucose level. Glucose enters cells for respiration. Excess glucose is stored as glycogen in the liver, and this process is regulated by insulin and glucagon hormones from the pancreas. In short, the text highlights two crucial homeostatic mechanisms in the body: maintaining a constant internal temperature and regulating blood glucose levels. Both utilize negative feedback to maintain balance. This image contrasts positive and negative feedback systems. Positive feedback intensifies physiological changes, moving further from the normal range. It continues until a clear endpoint reached, like childbirth. The example given highlights the extreme bodily changes during the labor and delivery, where the process accelerates until the baby is born. Conversely, negative feedback systems maintain homeostasis. They have three components: a sensor or receptor that monitors a physiological value, a control center that compares the value to the normal range, and an effector that reverses deviations to return the value to the normal range. The key difference is the positive feedback amplifies change, while negative feedback counteracts change to maintain stability. The image effectively illustrates these opposing mechanisms and their roles in bodily functions. This image describes childbirth as an example of positive feedback. At full term, the body doesn't maintain its existing state. Significant changes are needed to expel the baby. The process is self-amplifying. Once it begins, it must progress rapidly to a conclusion, otherwise the mother and the baby are at risk. The diagram shows this feedback loop. The baby's head pressing against the cervix sends nerve impulses to the brain, stimulating the pituitary gland to release oxytocin. Oxytocin is carried in the bloodstream to the uterus, causing uterine contractions which push the baby further against the cervix, repeating the cycle and intensifying until the delivery. This rapid escalating process is characteristic of positive feedback and systems.